Welcome to the Deep Dive. Today, we're embarking on an incredible journey. We're going microscopic to explore maybe the most intricate organ you have, the human eye.
It really is something else.
Yeah, forget any fancy camera you've seen. The eye is just light years ahead. It tracks motion, adjusts focus instantly. It even, repairs itself.
And that 3D world it creates, taking two slightly different images and seamlessly merging them. That's stereopsis. It's fundamental to how we perceive depth.
Absolutely. So our mission today is to really peek behind the curtain, dive deep into the microscopic stuff that makes it all tick. We've been digging into Histology, a text and atlas, 8th edition.
Great resource.
It is. We want to pull out the surprising bits, the crucial insights, show you not just what the eye is made of, but why it's designed so brilliantly. We'll look at layers, cells, how they work together, and even, what happens when things go a bit wrong clinically.
It's a masterclass in biological engineering, truly. Its main job, fundamentally, is taking light, focusing it incredibly precisely, and then turning that light into electrical signals your brain reads as sight.
Right, capturing the image. But like you said, it's more than that. That 3D thing, stereopsis, that's how you know how far away your coffee cup is.
Exactly. Gives us that depth perception.
And all this packed into something roughly the size of a ping pong ball, about 25 millimeters across. Held in place by tiny muscles, cushioned by fat. It's amazing.
Perfectly positioned, incredibly sensitive, and it's built in layers, kind of like a very specialized onion, the eyeball itself. 3 main coats concentrically arranged.
Okay, 3 coats. Let's start outside and work our way in.
Right. The outermost is the corneoscleral coat. Think of it as the eye's tough protective armor. Up front, you've got the transparent cornea, the clear window letting light in.
That's the part you see through.
Exactly. The rest of that outer layer is the sclera, the white part of the eye. It's dense, fibrous connective tissue, super strong, gives the eye its shape, anchors those muscles that let you look around.
So the white part isn't just passive, it's structural.
Okay.
What's the next layer in?
That's the middle layer, often called the uvea, or the vascular coat. And what's striking here is its color, usually a deep, dark brown.
Why it's so dark?
It's packed with blood vessels, providing nutrients, but also melanin pigment. That melanin is crucial. It absorbs stray light bouncing around inside the eye, cutting down glare, kind of like the matte black paint inside a camera.
Ah, smart design. Stops things getting fuzzy.
Precisely. And within this uvea, this middle layer, you have 3 main parts. The choroid, which is the main blood supply, especially for the retina. The ciliary body, which is key for focusing. And the iris.
The iris, that's the colored part, right? Blue, brown, green.
That's the one. It acts like the diaphragm in a camera, controlling how much light gets through the pupil.
And then the innermost layer. That must be the retina.
Yep, the retina. This is where the light-sensing magic happens. It's essentially the eye's film or sensor chip, packed with photoreceptor cells and complex neural networks. It's actually brain tissue, an extension of the central nervous system connected via the optic nerve.
Wow, so it's literally part of the brain sticking out. Okay, so we have layers.
Yeah.
But how does light travel through all this? You mentioned chambers.
Right. Thinking about the spaces helps. There are 3 main chambers. The anterior chamber is between the cornea and the iris. Then the posterior chamber, a smaller space, is between the iris and the lens.
Okay, front spaces.
And then the big one, the vitreous chamber, fills the whole back of the eye behind the lens right up against the retina.
And these chambers aren't empty, are they? They're filled with something.
Exactly. Filled with what we call the refractile media, 4 transparent things that bend or refract light to focus it. First, the cornea itself does most of the heavy lifting, the initial bending of light.
Okay, the cornea starts the focusing.
Then you have the aqueous humor. It's a watery fluid filling those front 2 chambers, the anterior and posterior. It helps with focusing, sure, but critically, it delivers nutrients to the cornea and the lens.
Why is that critical?
Because, surprisingly, the cornea and lens don't have their own blood vessels. They rely entirely on this aqueous humor for nourishment.
No blood vessels. That makes sense for transparency, I guess.
Absolutely. Then, behind the iris and aqueous humor, you've got the lens. It's clear, biconvex, curved on both sides, and crucially, it's elastic. It can change its shape.
That's for focusing close-up versus far away.
That's exactly it. Accommodation allows you to shift focus. And finally, filling that large back chamber, the vitreous chamber, is the vitreous body.
What's that like?
It's a clear, jelly-like substance.
Yep.
Mostly water, but with some collagen and other molecules holding it together. It acts as a shock absorber for the retina and helps maintain the eye's round shape.
So cornea, aqueous humor, lens, vitreous body.
Yeah.
Light passes through all four to hit the retina.
That's the path. And this whole intricate setup doesn't just pop into existence. It develops in the embryo through this incredible orchestrated process involving different tissue types coming together just right.
Okay, so that's the big picture anatomy. Now let's really zoom in, get down to the microscopic level, starting with that front window, the cornea. It's super thin, right?
Yeah, only about 0.5 millimeter thick in the center, but its structure is absolutely key to it being perfectly clear. The very outer layer is the corneal epithelium.
It's epithelium, like skin.
Sort of, but specialized. It's a protective layer, several cells thick. And what's amazing is how fast it regenerates, completely replaces itself in about a week. That's why a tiny scratch usually heals so quickly.
Okay, fast healing. Good feature.
And it's packed with nerve endings, makes it incredibly sensitive. You know, if you get even a tiny speck of dust in your eye.
Yeah, you feel it immediately. Very sensitive.
Exactly. And the stem cells for that constant repair. They're tucked away right at the edge of the cornea, where it meets the sclera, a zone called the corneoscleral limbus, like a dedicated repair crew on standby.
A repair crew at the edge. Fascinating. And you mentioned UV protection earlier.
Right. Our source material points out something really interesting. Unlike skin, which uses melanin, the cornea uses something called nuclear ferritin to help block UV light. This probably helps maintain its clarity.
Wow, okay. Different strategy for the same problem. What's under that epithelium?
Next is Bowman's membrane. It's a thin, dense layer, good barrier against infection spreading deeper. But here's the catch. If Bowman's membrane gets significantly damaged, it doesn't regenerate.
Uh-oh. What happens then?
It forms an opaque scar. Can permanently affect vision if it's in the central cornea.
So, handle with care. Then the main thickness of the cornea.
That's the corneal stroma, or substantia propria, makes up about 90% of the thickness. And this is where the magic of transparency really happens. It's made of, like, 60 ultra-thin layers, or lamellae, of collagen fibrils.
Collagen, like in tendons.
Similar building block, but the arrangement is totally different and incredibly precise. Within each layer, the tiny fibrils are perfectly parallel, but each layer is stacked at right angles to the layers above and below it, like microscopic plywood.
Plywood? Okay, crisscrossing layers. Why?
It's the perfectly uniform spacing of these tiny fibrils and the precise arrangement of the layers regulated by other molecules like proteoglycans that allows light to pass straight through with minimal scattering. That's transparency.
So if that perfect spacing gets messed up.
Exactly. If the stroma swells, say, from injury or disease, that perfect lattice gets disrupted and the cornea turns cloudy, opaque. And importantly, the stroma has no blood vessels, another key factor for clarity.
Makes sense. What are the final layers?
Deeper still is Descemet's membrane. It's actually the basement membrane for the innermost layer. Unlike Bowman's membrane, Descemet's can regenerate if injured, and it tends to get thicker as we age.
Okay, one regenerates, one doesn't. Got it. And the very inside?
That's the corneal endothelium, just a single layer of thin, usually hexagonal cells. But they are incredibly important. They act like tiny pumps.
The pumps. Pumping what?
Pumping excess water out of the stroma and back into the aqueous humor. They actively regulate the stromal hydration, keeping that perfect spacing we talked about.
So they keep the cornea from getting waterlogged and cloudy.
Precisely. If these endothelial cells are damaged or die off, and they don't really multiply much in adults, the pump fails, the stroma swells, and the cornea loses transparency.
That sounds serious. What happens then?
Significant endothelial cell loss often leads to corneal edema and necessitates a corneal transplant to restore vision. It's a very delicate balance they maintain.
Wow, all that complexity in just 0.5 millimeter. Okay, so that covers the clear part.
Okay.
What about the sclera, the white part? What's its microscopic story?
The sclera is much simpler structurally. It's dense, irregular connective tissue, mostly collagen fibers, but arranged much more randomly than in the cornea, which is why it's white and opaque, not clear. Its main job is toughness, protection, maintaining shape, and providing attachment points for those eye muscles.
It connects to the cornea at that limbus zone we mentioned.
Exactly. That transition zone, the corneoscleral limbus, where the clear cornea meets the white sclera. And remember, that's where those corneal stem cells live. It's also near where the eye's internal fluid drains out.
Yes, the drainage system for the aqueous humor. Can you walk us through that again? How does that work at a micro level?
Sure. So the aqueous humor is made by the ciliary body, flows forward through the pupil into the anterior chamber. Then in the angle where the iris meets the cornea, the iridocorneal angle, there's this spongy network called the trabecular meshwork.
Spongy network.
Yeah, think of it like a sieve. The aqueous humor filters through the tiny spaces in this meshwork and then flows into a circular channel running around the eye called the scleral venous sinus, or canal of Schlemm. From there, it drains into the bloodstream. It's a constant production and drainage cycle.
Keeping the pressure just right.
Ideally, yes, maintains the intraocular pressure.
But what if that trabecular meshwork gets clogged or doesn't work efficiently?
That's the core problem in the most common type of glaucoma, open-angle glaucoma. The drain is open, but it's not draining well enough. Fluid backs up, pressure inside the eye rises.
And that pressure damages the optic nerve.
Exactly. It compresses the delicate nerve fibers where they exit the back of the eye. Over time, this damage leads to loss of peripheral vision first, and eventually, if untreated, can cause blindness.
Scary, but treatable if caught early.
Yes, very much so. There are eye drops to reduce fluid production or improve outflow, laser treatments to open up the meshwork, and surgical options too. Early detection is key.
Good to know. Okay, let's move inward again to that middle vascular layer, the uvea. We mentioned the choroid supplying blood, but let's talk iris and ciliary body. The iris, it's not just about color, is it?
Not at all. The color comes from pigment cells, melanocytes in the front part, the stroma. Fewer melanocytes, more light scattering, you get blue eyes. Lots of melanocytes absorbing light, you get brown eyes.
Simple physics, basically.
Pretty much. But its main function is controlling the pupil size. It has two tiny muscles. One is circular, the sphincter pupillae. When it contracts, like in bright light, the pupil gets smaller.
Let less light in. Makes sense.
Then there's the dilator pupillae. Its fibers run radially, like spokes on a wheel. When they contract, usually in dim light, they pull the pupil open, making it larger.
Letting more light in. So
Yeah.
one constricts, one dilates. Constantly adjusting.
Constantly. It's an autonomic reflex, happens without you thinking, controlled by different parts of your nervous system.
Okay, behind the iris is the ciliary body. You said it's involved in focusing.
Yes, it's a ring of tissue. Inside it is the ciliary muscle. It's actually quite complex, with fibers running in different directions.
Yeah.
When this muscle contracts,
Yes.
it actually relaxes the tension on the tiny fibers holding the lens. This allows the elastic lens to bulge, become more rounded.
More rounded for seeing things up close.
Exactly. That increases the lens's refractive power for near vision. When you look far away, the ciliary muscle relaxes, which increases tension on the lens fibers, pulling the lens flatter for distance focus.
That's accommodation. The eye's autofocus. Amazing.
It really is. And the ciliary body also has these little extensions, like tiny ridges, called ciliary processes. They stick out towards the lens.
What do? They look important.
They are super important. 3 big jobs. 1, they are the factories that produce the aqueous humor.
Ah, the fluid that fills the front chambers.
2, their specialized epithelial cells form the blood-aqueous barrier. Tight connections between these cells prevent unwanted stuff from the bloodstream getting into the eye's fluid. Keeps the internal environment stable.
Like a security checkpoint.
Good analogy. And 3, they secrete and anchor the zonular fibers, those tiny transparent ligaments that hold the lens in place and transmit the force from the ciliary muscle.
Wow. The ciliary body is a real workhorse. Making fluid, controlling barriers, holding the lens, and focusing.
It does a lot. And behind it, lining most of the back of the eye, is the choroid. Again, that dark, vessel-rich layer nourishing the outer retina.
Right. Which brings us finally to the retina itself, the light sensor. But also quite fragile, it seems. You mentioned retinal detachment.
Yes, that's a critical issue. The retina has 2 main parts conceptually. The neural retina, which contains the photoreceptors and neurons, and the retinal pigment epithelium, or RPE, a single cell layer it rests on.
Okay.
Normally they're held together, but if they separate, maybe due to trauma or age-related changes in the vitreous jelly pulling on it, that's a retinal detachment. The neural retina gets separated from its nutrient supply via the RPE and underlying choroid.
What are the symptoms?
People often describe a sudden increase in floaters, maybe like a shower of black dots, flashes of light, or a shadow or curtain seeming to fall across their vision. It's a medical emergency.
Needs quick treatment to save vision.
Absolutely. Often laser surgery or other procedures are used to reattach the retina and seal any tears.
Okay, so keeping the retina attached is vital. Now, within the retina, are all parts the same?
No, definitely not. There's the optic disc, or optic nerve head. That's where all the nerve fibers bundle together to leave the eye and go to the brain. There are no photoreceptors there at all.
The blind spot.
That's the one. But then very close to it is the macula lutea, a small yellowish area. And right in the center of the macula is a tiny pit called the fovea centralis.
The fovea. You said that's for sharp vision.
The absolute sharpest. It's packed almost exclusively with cones, the photoreceptors for color and fine detail. When you look directly at something to see it clearly, you're aiming your fovea at it.
Makes sense.
Right.
And this is the area affected by macular degeneration.
Yes, age-related macular degeneration, or ARMD. It's the leading cause of significant vision loss in older adults in developed countries. It damages the macula, particularly the fovea, destroying that sharp central vision needed for reading, driving, recognizing faces.
Devastating. Is there just one type?
Primarily two forms. Dry ARMD involves the gradual breakdown of the RPE and photoreceptors, often with deposits called drusen forming. Wet ARMD is more aggressive, involving abnormal blood vessels growing under the retina, leaking fluid and blood, causing rapid damage. There are treatments, especially for wet ARMD, but no cure yet.
Okay. Now, the retina itself, I heard it has layers, like a lot of layers.
It does, classically described as having 10 distinct layers visible under a microscope. It's basically a highly organized stack of specialized cells and their connections.
10 layers.
Yeah.
Wow. Can we hit the highlights? What are the key players?
Definitely. Starting from the very back, right against the choroid, is that crucial retinal pigment epithelium, the RPE layer we mentioned.
Okay, the support layer.
Then comes the layer of photoreceptors themselves, the outer and inner segments of the rods and cones. This is where light detection actually happens.
Rods and cones. Got it.
Then you have layers containing the cell bodies, nuclei, of these photoreceptors, and layers where they make connections or synapses with the next set of neurons, bipolar cells, horizontal cells, amicline cells.
So layers for detection, layers for processing.
Exactly. There are layers containing the nuclei of these processing neurons, more layers for their synaptic connections, and finally the ganglion cell layer. These are the output neurons.
Output, meaning?
Their long axons bundle together and form the optic nerve, carrying the processed visual signal out of the eye towards the brain. The innermost layers are these nerve fibers and a final limiting membrane.
It's like a mini computer built right into the eye.
That's a great way to think about it, a sophisticated processing network.
Let's talk about those RPE cells again. What exactly do they do? Seems like more than just support.
Oh, they're incredibly active. 4 huge functions. 1, absorb stray light that gets past the photoreceptors, preventing scatter. 2, form the outer blood-retina barrier, controlling what gets from the choroid's blood supply to the photoreceptors. Very selective.
Okay, absorption barrier.
3, they are essential for regenerating the photopigment molecules, like rhodopsin in rods, after they've been bleached by light. They recycle the crucial vitamin A component. And 4, they act like garbage collectors.
Garbage collectors.
Yeah. The tips of the rod and cone outer segments are constantly shedding old membranous discs. The RPE cells engulf and digest this shed material through phagocytosis, constant renewal.
Recycling and waste disposal. Busy cells. Now, the rods and cones themselves. Big difference between them.
Huge difference in function and location. Rods, about 120 million of them, are incredibly sensitive to light. Responsible for night vision, seeing in shades of gray. They're found throughout the retina, except in the very center of the fovea.
So rods for dim light, black and white.
Right. Cones fewer, maybe 7 million, need more light to function, but give us color vision and sharp detail. They're most concentrated in the fovea, which is why central vision is so sharp and colorful.
And there are different types of cones.
3 main types, each containing a slightly different light-sensitive pigment, iodopsin or photopsin, that responds best to different wavelengths, roughly corresponding to blue S cones, green M cones, and red L cones. Your brain compares the signals from these 3 cone types to perceive the full spectrum of color.
So how does the light actually become a signal? You mentioned something counterintuitive earlier.
Yes. It's fascinating. Inside a rod or cone, there are molecules like rhodopsin in rods. It contains a form of vitamin A called 11-cis-retinol. When light hits it, it instantly changes shape to all transretinal.
A shape change, like flipping a switch.
Exactly. This shape change triggers a cascade of events involving other proteins. Now here's the weird part. In total darkness, the photoreceptor cell is actually partially depolarized and constantly releasing a neurotransmitter called glutamate.
Releasing a signal in the dark.
Yes. But when light triggers that shape change and the subsequent cascade, it leads to the closure of certain ion channels. This makes the inside of the cell more negative. It hyperpolarizes. And this hyperpolarization decreases the release of glutamate.
So light causes less signal to be released.
Precisely. It's the reduction in glutamate release that the next neuron in line, the bipolar cell, detects as a signal that light has been perceived. It's an off signal that means on to the brain, basically.
Mind blown. It's completely backwards from what you'd expect. And then the RPE has to reset that retinal molecule.
Yep. The RPE cells and some other supporting cells are crucial for converting that all-transretinal back to 11-cis retinal so it can be slotted back into the opsin protein, ready to detect another photon of light. The cycle repeats constantly.
And if someone has a problem with one of their cone types, say the red ones,
Then they'd have red-green color blindness, specifically protanopia. Difficulty distinguishing reds and greens because the L cones aren't working right. Deuteranopia involves the M cones, green, tritanopia, the S cones, blue. Often genetic and more common in males because the genes for red and green pigments are on the X chromosome.
makes sense. Okay, moving away from the retina for a moment. The lens, we said it focuses light and changes shape. What's it actually made of?
It's unique, transparent, avascular, no blood vessels, and no nerves. It's enclosed in a capsule, like a clear elastic bag. Inside, just under the front part of the capsule, is a single layer of epithelial cells.
Okay.
These epithelial cells, especially near the lens equator, are constantly dividing. The new cells elongate dramatically, lose their nucleus and other organelles, and fill up almost entirely with special proteins called crystallins. These elongated cells become lens fibers, packed together in perfect layers like an onion.
Losing organelles, again for transparency.
Exactly. Anything that might scatter light is eliminated. It's the high concentration and precise arrangement of these crystalline proteins that makes the lens transparent and gives it its refractive power.
But it gets less elastic with age.
Yes, that's presbyopia. The lens hardens, the crystallines might change slightly, making it harder for the lens to round up for near focus, hence reading glasses.
And cataracts.
That's when the crystalline proteins clump together or change in ways that make the lens cloudy, opaque. Vision gets blurry, colors seem faded. Very common with aging, but also caused by things like diabetes or UV exposure. Thankfully, cataract surgery is very effective. Remove the cloudy natural lens, replace it with a clear artificial one.
Amazing technology. What about the vitreous body, the jelly in the back?
Right, fills that big vitreous chamber. It's mostly water, about 99%, held in a gel-like state by a network of collagen fibers and hyaluronic acid. There are a few cells called hyalucytes in there too.
And its job again.
Mainly, shock absorption protects the retina during movement and maintaining the eye's overall shape. It also helps keep the neural retina pressed against the RPE for proper function.
Okay, so we've covered the eyeball itself in incredible detail. What about the structures around the eye? Eyelids, lashes, tears?
Crucial accessory structures. Eyelids are basically folds of skin reinforced by a dense connective tissue plate called the tarsal plate. They protect the eye, spread tears, and contain muscles to open, levator palpebrae, and close, orbicularis oculi. Eyelashes help keep debris out.
And they have glands too, right? I've heard of styes.
Yes, several glands. Tarsal glands, or meibomian glands, are large sebaceous glands in the tarsal plates. They make the oily outer layer of the tear film, which stops tears evaporating too quickly. If one gets blocked and inflamed, you get a chalazion.
Okay, chalazion.
Then there are smaller sebaceous glands, Zeiss glands, associated with the eyelash follicles. An infection there causes a stye, or hordeolum, plus some small sweat glands, Moll glands.
And the tears themselves, where do they come from?
The main lacrimal gland is tucked up under the outer part of your upper orbital bone. It produces the watery component of tears. Tears then flow across the eye, drain into tiny openings called puncta at the inner corner, go through little canals, canaliculi, into the lacrimal sac, and then down the nasolacrimal duct into your nose.
That's why your nose runs when you cry.
Exactly. And tears aren't just water. They contain lubricating mucus, oils from the meibomian glands, salts, and importantly, antibacterial proteins like lysozyme and antibodies. They clean, lubricate, and protect.
A complex protective fluid.
And
finally, the eye muscles.
6 extrinsic muscles attached to the outside of each eyeball, 4 rectus muscles, superior, inferior, medial, lateral, and 2 oblique muscles, superior, inferior. They work together with incredible precision, controlled by 3 different cranial nerves, to move your eyes rapidly and accurately, track objects, and keep both eyes pointed at the same target. Essential for stable binocular vision.
Okay, wow. So, stepping back from all this incredible microscopic detail, what's the big takeaway?
I think it's the sheer complexity and honestly the elegance of the design. We started comparing it to a camera, but it's so much more. It's a living, self-repairing, adaptable biological system operating with incredible precision, from the molecular level of light detection right up to the coordinated movement of the entire globe.
The level of integration is just staggering. Everything has to work perfectly
It
together.
really does. And thinking about this intricate physical machine, it really makes you ponder perception itself, doesn't it?
How so?
Well, if the very mechanism of seeing involves all this complex filtering, processing, even counterintuitive signaling, like light decreasing a signal, how much is our perception of the world actively constructed by this system rather than just being a passive recording?
Is seeing truly believing when the believing part is built on such a complex, active biological process?
Exactly. It makes you wonder what other seemingly simple senses might hide equally intricate perception-shaping mechanisms just beneath the surface, waiting for us to explore.
That is a fantastic thought to end on. A reminder that even the things we take for granted are often universes of complexity. We really hope you enjoyed this deep dive into the eye's microscopic world.